Welcome everyone, welcome class. I'm glad you could join this session. This video actually is to introduce you to the fundamental skills that you will need throughout our future practical works in immunology. So whether you are watching this after our face-to-face session in class at the University of Bamenda, or if you are just catching up at home, I want you to treat this as your go-to reference guide before every single practical session. Please don't hesitate to come back to watch the video anytime before a new practical session. So let us get started. Okay, here is the plan of our today's session. We will cover these 10 topics that are organized in logical order, from how science works all the way to how you document what you discover in the lab. So how to write a proper scientific lab report. Let's start with the first. The scientific method is the foundation of all scientific work. Before you touch a single piece of equipment in a lab, you need to understand what is the method you have to use. You need to understand the cycle of inquiries that belongs to the scientific method, that compose the scientific method. Okay, we used to say that science is iterative. So why do we say that? Because we used to think science is a straight line in the past, because you have a question and you want to find the answer. To reach to the answer, you have to apply some precise steps. It was a bit mechanical, so you just have to check, use a particular method, apply that method, and get to the answer. But at the end of the day, your answer will lead to new questions, and you will continue with new experiment. That's why scientific method is considered as science itself is considered as iterative, because it's a cycle. You start by making observation, you generate hypothesis, make prediction. Prediction is what result you expect to see, and from that you design and conduct experiment, and finally you have your answer, and from this answer you will surely have new questions. This is how you construct a scientific model. First, it's a model that is sure that will lead you to the answer if you ask this type of question, or if you made this observation, and from that we can say the scientific method is a cycle. But let's see how it has been developed in the precise immunology context. So the birth of vaccination history will indicate actually how we get to the scientific method in immunology that we are using today in the lab. So it starts with Edward Jenner from England. It was a country physician. So he made an observation in the past. He made an observation because he was paying close attention to the people around him. So he observed that milkmaid, the young women that were taking care of cows every day, they seems to be protected from smallpox. Smallpox was one of the most feared disease of the era at the time, because it was disfiguring people. It was frequently leading to death. So it was responsible for killing 400,000 Europeans every year at that time. So, but milkmaids were untouched by the disease, and their skin were the only ones that kept their, let's say, their shining. And there was an ideal of beauty at that time, that the complexion of a milkmaid, because they appear to be untouched by the disease. And Jenner did not assume that it was a superstition, like milkmaids were protected from smallpox. He just observed. He was observing them, and he notes that these same women frequently contracted cowpox, because they were dealing every day with cows. with cow. So the cow have a mild form of the disease, mild form of the disease. The cow from the infected cattle, that was cowpox, sorry for the pronunciation. So the cowpox caused a local pustular lesion on their hand, a brief fever, and then after that they just fully recovered. So it was unpleasant, but it was benign compared to smallpox clinical features. And after suffering cowpox, these women appear immune to smallpox. They were immunized because they've never been infected by smallpox. So now here is where the scientific method begins. Because Jenner did not stop at just observing and noting that okay, these women, when they have cowpox, they never developed smallpox. No, he made an observation first, and then he formulated an hypothesis. Exposure to cowpox confer protection against smallpox because for him both were associated. So he reasoned that something in cowpox material, something that probably is from biologic structure of cowpox, was responsible for the protection, and that is something that could perhaps be deliberately transferred from person to person or from animal to person. So he designed and carried out his critical experiment that can be, we can never reproduce it today, because he used first a milkmaid named Sarah. He collected cowpox from her cowpox lesion on her hand. He collected fresh material and he took the material, the fluid from the pustules, and he inoculated that into a small cut on the arm made on a 8 years old boy named James. So the son of James was the son of his gardener. The boy developed mild fever, small local reaction, small pustules, but he recovered completely after that. So 6 weeks after, he now collected fluids from an infected person with smallpox, and he did repeat the experiment by inoculating it to the same small boys. But after that, the small boy didn't develop any infection, just mild fever and recovered. So he tried again 6 months after, same result. He concluded that okay, this was the cowpox that protects the little boy from smallpox, and he named this procedure the vaccination, from the Latin vacca, that means cow. He submitted his finding to the Royal Society of London, but that was rejected at that time, so he published it in a private review. So the observation here was that milkmaid who had the cowpox first time will never develop smallpox. So the hypothesis was the cowpox infection induced protection against smallpox. So for him, the two diseases were sharing something biologically relevant. And he predicts that if he infects in first time with cowpox in small boys, the small boys will not be infected with smallpox. So that means he will not develop the disease, and that was in the majority of the case leading to death. So the experiment was inoculation of this little boy. That was controlled in the sense that Jenner followed up with a deliberate smallpox challenge to test the predicted protection. So the model that eventually emerged from Jenner's works was the vaccination model. This was the first time that they were using another disease to protect from another disease. So, but it was not the first to use the techniques, but the methods to use a small, let's say, a small or light form of a disease to protect from the most dangerous form of disease, he was the first, because in the past, like many centuries before, in China, the practice was used, since, but it was named variolation because they were using few quantity also of pustule to protect from the smallpox. And in Africa also, variolation was practiced independently across part of the continent. It reached Boston with slavery, and yeah, it has been reported in some. This technique has been reported from that time. Also, we have some mention of Lady Mary Montagu, the wife of a British ambassador in Constantinople in Turkey. The Turkish population, they were using that techniques, like taking few fluids material from an infected person to infect a normal person so that they can prevent them from getting a really harmful form of the disease. So they used to do that. So we know today, we know the contribution of Jenner was not the first when we are talking about the technique, but he was the first to discover the protection against smallpox coming from cowpox. So he demonstrated that a related safer animal virus, that was cowpox, could substitute a smallpox material, eliminating the risk of causing the very disease one was trying to prevent. So that was a real scientific advance at that time. And Jenner had no concept of B cells, T cells, or antibodies. He did not know at that time what a virus was. He was working almost a century before Pasteur and Koch established the germ theory of disease. So it was just observation, generating hypotheses, making prediction, and yeah, conducting experiment to prove if the hypothesis was true or false. So this is why I tell you the scientific method is more powerful than any individual piece of equipment in the lab. Jenner had a lancet. He just had a lancet, a fluid coming from the biological material of infected person, and one hypothesis he want to try. What he built with those tools, these basic tools, actually changed the course of human history. Immunology, it's changed the course of immunology. So when you walk into the lab and set up your agglutination test or your complement system test, I say you are working within the same tradition. Observe carefully, hypothesize precisely, and design your control thoughtfully. Record your result honestly, and when the data surprise you, do not discard it. That surprise might be the next Nobel Prize. So the surprise might be the most important observation you make. So always be honest with the result, and also the reporting should be like what you observe is what you report. Don't invent anything, don't create anything, don't try to make your result look more fancy or more, I don't know, astonishable. Just write what you observe, how you observe it. Okay. Okay, now we are done with the theories. Let's start with the practical session. Practical exercise are where theory becomes reality. So if in the following the next slide, we will develop that. We will talk about what you should expect and what skills you will develop during our future practical session in the lab. Okay, this semester we have 4 experiments to cover, 4 practical sessions. The first one is complement fixation test. Second, agglutination reaction. The third one will be radial immunodiffusion, and the last one, dot ELISA. So before each practicals, you have to read the entire protocol from start to finish. Please don't read just the procedure. Read the full entire protocol. This is not an option. It will prevent from errors in the lab. It will save time, and most importantly, it will keep you safe in the lab. So please do your job as a student and read the handout before coming in the lab. So here you have skills that has been listed on the slide. So there are skills that exactly what your practical report will be assessed on these following skills. So if you follow the instruction carefully and in the correct order, if you handle the laboratory apparatus correctly and safely, so if you take care of the equipment in the lab, having due regard for safety at all times, from the moment you enter in the lab to the moment you go out, you need to keep on your API. So making accurate and reproducible observation, yes, discuss this in group. Record results in the most appropriate form. Please, we will introduce you the different form to be used when it's about reporting the result in the next slide. Present quantitative result clearly. So how to present a quantitative result, we will also define that and clarify that. Drawing valid scientific conclusion from experiment data, writing a professional scientific laboratory report. So here are the key points that will determine the marking guide. Okay, they are the marking guide actually. Okay, following instruction. This might seem obvious, but in a laboratory setting, failure to follow instruction is the single most common cause of failed experiment and accident also. So be really careful and serious about that. A very common mistake among students is to start working before reading the full protocol. I will repeat over and over, please read the entire protocol before coming in class, in the lab. So then some of you, they don't read the protocol, then midway through the experiment, they will discover a step that requires something they did not prepare, and this is where you start failing the experiment. I always advise read the protocol the night before, even 2 days before the practical. Highlight unfamiliar term, search definition if you are not aware of a material. If the name of that material don't appear, please search for an image or a video on YouTube that will present you what is this material if you don't know it. So look them up. Come to the lab prepared. The example on the left and right panel here will show you the mindset, the preparation before and discipline during the experiment in the lab. So before you start, read the protocol. I repeat it again. Read the entire protocol. Understand the purpose of each step. Before acting, you need to understand why we are doing that. What is the purpose of this particular step, and why are we using this material? What is it used for? Gather all material before starting. So you have to have your list. Tick on your list if you have each of the material that has been listed there. Do our groups have everything on our table before we start? Ask your instructor if anything is unclear regarding the protocol, the procedure, or a material. If you don't know, please ask. We are there for that. Never attempt an experiment without a lecturer's knowledge. So you have prerequisites that you need to know before starting an experiment. Please take the time to read about that. Take the time to make some research before doing an experiment. And now, during the experiment, follow number step in strict order. Don't skip or rearrange the steps. Record data as you go, never from memory. Don't trust your memory. Even if it's for 2 seconds, please write it down. Even if it's on your hand, actually your hand should have gloves. So write it on a paper. You should have this small paper tips that you should have in the lab to write the different data on it. Please don't trust your memory. Even if it's just for the time for you to find a pen, you can forget or you can mix, because this is how our brains work. So don't trust your memory. Record the data as you go. If something goes wrong, stop and inform the instructor. And also be time sensitive on the steps demand full concentration. So you should have and be in the future. So you should be aware of what time or approximately the amount of time one step can take. If you have something to prepare in between 2 steps or while waiting for one step to finish, you should be aware of the time of each step before, and this is during the experiment. So if you took the time to read the protocol, the entire protocol, you should know what it can take as amount of time. Okay. And the section 4 is handling apparatus. Because biological science require precise use of specialized equipment. So let us review key apparatus you will encounter in our immunology practicals in the futures. Just few of them. First, the microscope to inspect cells to ensure slides are clean and lens smear free. So each piece of equipment has a specific correct way to be handled. Misuse can damage the equipment, invalidate your result, or it can also cause injury. So for micropipettes, for example, the most common error is using the wrong volume setting or not ejecting the tips between 2 samples. This is common error we are doing every day. You can just because you are in a hurry, you collect the pipette from your neighbor, and this is how you just miss to readjust the volume you need, and you use the same volume when you are not supposed to use that volumes. Okay, so always be concentrated, be focused. Make sure you have your material. You check the material every time before using it, and if you are concentrated, if you are focused, you will remember to change the tips between 2 samples. For centrifuge, an unbalanced rotor is a serious safety hazard. That is also a common mistake, to don't balance the centrifuge. So pay attention. In the next practical, that will be complement fixation practical, you will use a water bath. You will use a water bath, and you need to make sure it is set to exactly at this exact temperature that is mentioned in the protocol because it is 56 degrees Celsius before placing your serum in the sample. For this experiment, you need to respect, because the temperature is critical for inactivating the complement system in the blood. So you need to respect strictly the temperature. Okay, this is just few examples. Okay. So in the next section, having due regard for safety, always safety first. The lab is a safe environment, but only when everyone behaves responsibly. You need to be responsible. Let us go over the non-negotiable safety rules. Here are the rules. The dress code. At the University of Bamenda, we take laboratory safety extremely seriously. So the most critical role in immunological practical is the handling of human blood. And you know, in immunology, we are using a lot of human blood. So in practical 1 and practical 2, where we are going to cover the complement and the agglutination test, you will be using your own blood or one classmate blood. So this must be done with gloves on, and any lancet used or syringe must go directly into the sharp container. Don't play with that. In the sharp container, a solid, really sharp container, okay? And I want to emphasize the body fluids rules. Never allow your blood to contact anyone else. This protects everyone during the experiment. So if you are unsure about any procedure, just stop what you are doing, stop everything, and ask. An injury because you did not ask is far worse than any delay caused by asking a question. So choose to delay the full class, but you need your safety need to prevail. So go through these following rules. The dress code, you need to have your lab coat, closed shoes, tied back hair at all times during practical. For body fluid, treat all human blood as a potential infectious. That means you need to use gloves all the time. For the chemicals, pills, if acid, if it's contact with your eyes or skin, wash immediately with water, good amount of water, and you need to inform the lecturer in the lab. Electrical equipment, never allow contact with water. Pay attention to that. It should not be in contact with water when we are doing the experiment. Also, no food, no eating, no drinking in the lab, no smoking for sure. When we are going to use the Bunsen burner, use paper to light it up. Allow equipment to cool before handling. Never leave unattended. And for the waste disposal at the end, solid waste in bins only. Lids on sharp container. All the sharp container that having syringes, all this thing with blood collection material, please, it has biohazard bags or biohazard sign on it. So make sure to dispose it in the right type of container. And no entry without permission. Wash your hand before you leave and throw your gloves, not your hand, you need it for later. So throw the gloves in the trash that is available at the exit of the lab. Okay, just trash it there. Wash your hand with soap, clean water before you go out of the lab. Okay. So making accurate observation, this because science is built on careful objective observation. So in our immunology practicals, the difference between what is a positive or a negative result will rely on that observation, and sometimes observation rely on it's just a subtle visual change. So here we will see how to accord to it. So many of our results are visual, as I was saying, like for agglutination, for precipitating rings, color development in ELISA. The skill of accurate observation is therefore particularly important. So for example, in the agglutination test, a positive result appears as fine red granule within 30 seconds. If you are not watching carefully, or if you read too early or too late, you might miss the result. So I often say a good scientist observe like a detective. So your observation, your visual ability should be you should be sharp with it. Every details matter. Don't be focused. So you don't miss a variation of color or fading. Okay. So and nothing should be assumed. Also for negative result, scientifically valuable. I don't know why when we have we are really very happy when we have positive results, but negative results are also, uh, I can't say as much important as positive result, because it can prevent you from wasting your money doing the experiment again if you are aware that this hypothesis, this method I apply lead to a negative result. So it can prevent others to waste their time and their money, and yeah, and it makes the science to progress also. So if there is no agglutination, that is still important information. Please just relieve that there is no agglutination. Don't try to at all costs have a positive result. This is not the aim of the practical session. So you need to make accurate observation. Know what you are looking for before starting. So that's why you need to know the prediction, what result you expect to see. Use a quantitative over a qualitative description whenever it's possible. It's not possible all the time, but when it's possible, please use quantitative. It's always more trustable than qualitative. Yeah, so record the exact time of observation. Many immunological reactions are time dependent. So you need that. When it's time dependent, record the time when you observe. Distinguish between what you observe directly against what you infer. If using a microscope, clean lens first. You don't want to confuse or to identify something as a leukocyte when it's just a dust. So use calibrated eyepiece to measure cells and repeat observation. Do not rely on a single reading, because in biological we always have you can have triplicate. This is why you have biological replicate. So it varies, and for negative result, they are still result. So record them accurately. Okay. So the next section is the recording result in an appropriate form. So good science is reproducible science. That is only possible if results are recorded properly. So this is how we record results, the tools and rules. For quantitative, this is numerical, the type of data. You can have quantitative form of data. Qualitative, that means it's just descriptive, like you say positive, negative, and when it's quantitative, you have precise number. When it's semi-quantitative, this is where you have, for example, grade, scale for hemolysis, for example. So here in the table, you can see an example of the blood grouping tables, how to report the result. If negative for anti-A, positive for anti-B, then the blood is group B. So these scientific drawings here, you have to cover, it has to cover at least 1/3 of an A4 page. Plan proportion before drawing. Don't start drawing tables and you have the end of the table in another page. Make sure everything will fits in one page. Also use sharp HB pencil if you have to draw or to sketch your result, so you can draw clean and firm lines, so no shading unless necessary. When you use a microscope, do not draw individual cells for low power plants. Draw boundaries only, and always include a skull bar. Always a skull bar, and label all the structure very clearly. At least on the first, the control, what refer to the control drawing, we need to label it, so that for the rest we have a marker, we have something to compare with, we know exactly what is done for. Okay. So now the presentation of quantitative result, number alone do not tell a story. So you have to display your data to determine whether your findings are understood. Okay, the most important graph for our immunology practicals are scatter plot. You can see on figure 1.4 what the scatter plot look like. particularly for practical number 3, that will be focused on radial immunodiffusion, where we will plot precipitating ring diameter squared against antigen concentration to create a standard curve. So when we construct this graph correctly on linear paper with units, proper label axis, we can read of unknown concentration with confidence. So we rely on this axis, on this linear, the line to found unknown concentration. This is how we have to be really careful and really exact about our when we are drawing. So a poorly drawn graph is worse than no graph. It can mislead the reader and undermine the scientific credibility. So here is the right graph type you should use. You can use a line graph that if you have a continuous data over the time, like for example antibody titer over weeks post vaccination. The second type is the bar chart, when we compare discrete categories like blood groups and distribution in a cohort. In the third, we have frequency distribution. So, for example, distribution of ring diameter in radioimmunodiffusion assay. And the last one, we were talking about the scatter plot relationship between two variables, antigen concentration against ring diameter. Okay, so we have some rules regarding graph construction. First, we need to plot the independent variable on x-axis and dependent variable on y-axis. We have to label both axes fully, including units. So we need to write it down, all the meaning of the axis, of the name. Don't use abbreviation or something like that. No, put the name and the unit in bracket. Use a ruler. It's always better with a ruler, is more precise. Start axis at 0 unless scientific convention dictate something else. And in particular radioimmunodiffusion assay, we plot diameters against concentration, squared diameters against concentration on linear paper. So this will give a straight line for interpolation. You have to interpolate, to extrapolate actually, to have the concentration. So it has to be exact. Include a figure legend below each graph explaining what is shown, what are the groups, the axis, what is represent, and everything. You need a legend. Okay, so this is how you should report your data in our future practicals. One of these 4. Okay, so conclusions are where our data becomes knowledge. So this requires scientific reasoning, not just describing numbers. This is where you know if you understood the practicals. And the scientific reasoning, the conclusion section of our lab report, the lab report you will provide at the end of the experiment, is where you should demonstrate your scientific maturity. You should demonstrate that you understand. It's not enough to say the results support my hypothesis. No, you want to explain why the results support your hypothesis. So what biological mechanism underline the observation? This is you don't just confirm. And what are the limitations of your conclusion? So if you don't have all the data, all the elements to be able to provide a steady answer, just give the limitation. What inhibits you? What prevents you from confidently give an answer? For example, in the complement fixation test, if hemolysis do not occur, the conclusion should state the absence of hemolysis indicate that complement was fixed by the antigen antibody complex formed in the patient serum, suggesting the presence of a specific antigen a specific antibody to the test antigen. So this is how, for example, um, you can address a limitation or an unexpected result. You will read, because in that particular experiment we are focusing on pathogens. So the classical, not the classical pathway, but other pathway that are involving pathogens. So that's why I'm taking this example. So that is a scientific conclusion. Notice how it links to the observation. It's linking the observation to the mechanism. This is how you should produce conclusion. Good conclusions. It's how you should draw conclusion. Okay. State clearly whether the hypothesis was supported or refuted by your data. You have to say that clearly in the conclusion. Never say a hypothesis is proved, because science, we say science is iterative. Okay, so science produces evidence, not absolute proof. What you prove today can be disproved tomorrow. So site specific result. The agglutination was observed in NTA wells only, indicating blood group A. An example. Explain any unexpected result. Any unexpected result should be explained. Is it an effective limitation? Is it implication of another source of pathogen that you didn't mention? Is it protocol failure or something? Just explain any unexpected result, because these are scientifically valuable. It can indicate other researchers, if they want to redo your experiment. If it's something that is related to your SOP, you can just do it again. But if it's something that need a more or a deeper thought about the protocols, then they will redefine the protocol again. So discuss limitation of your experimental design honestly. Compare your findings with published literature. This is what scientists do. And suggest how the experiment could be improved or extended. This is always good to suggest how it could be improved, because it's giving to other a gap. You are just presenting the gap in the literature that other scientists can fill. So distinguish between correlation and causality. Correlation is when you have two variables that will change together, and causality is one cause the other. So the change of one is causing the change of the other. So this is where you have causality. They are linked. Okay. And the finality, your lab report is the permanent record of your experiment. It must be written so that another scientist could reproduce your work exactly. So someone can just follow what you said and reach to the exact same result you have. So the slide here is let's call it your cheat sheet, because for the lab report, so you have just to study it to know the different parts that need to be present in your final lab report. So the most common mistake I see is when some students are putting interpretation in the result section. No, the result should be described, should just describe the data and not explaining the data. In conclusion, some that do not refer to specific result, they are just concluding from nowhere, and this is not good. Conclusion, always anchor your conclusion in actual number of observation. And also missing references. I don't know why we don't like doing literature search. We don't like searching for bibliography and using right references. Don't bring me back the same reference that are in the handout. You have to search your own and add to it. Do your job and search more references so you can compare your result with it, and so it can help you to develop your critical thinking also. And for our course, I expect a report in APA format for references. If you are unsure, you have one video about the referencing, so go back to it and just read it, just watch it to have the presentation of the different format. Okay. So I have compiled this list from years of grading student lab report. These are the common mistake that cost marks uh, most of the time. So, for example, interpretation in the result section, never. Vague conclusion that are just one line, that are just saying that okay, my hypothesis was true or was false. No. Copying the protocol has method. No. Learn to be concise, precise, and be able to do the reporting, to use the reporting speech when it's about to describe the methodology. Don't just copy back the protocols procedure as it is listed in the handout. So no unit on the axis or in the table, the raw data without summarization. This is where you have to use graph. Don't put back all the single data from all the samples. No, you have to summarize it, group them, and produce either a graph or a scatter plot or something like that. No scatter plot. No negative control mention. Always mention the negative control. Even the technical control, mention them. Okay, so what you should do is for the result. You should describe exactly what's happened. The discussion is why it's happened. Distinguish between the two. Always reference specific numerical result in your conclusion. Write in past tense, the first or the third person. Describe what you did. Okay, and every number must have a unit. Summarize into means. Use table and figures, and always state and explain your control experiment. Explain it in the methodology. What is your control experiment? Okay. So in the result, for example, you will say the precipitating ring diameter was 12 millimeter, while in discussion you will say the ring diameter of 12 correspond to an IgG concentration of approximately blah 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 based on the standard curve. So you have to explain now what is the meaning of that. Okay, and so the key takeaway, we reached the end of this introductory lecture on practicals. So I want you to come to every practical session having reviewed these 10 principles. I will give you the PDF of this summary slide if you want. You can print it and also keep it in your lab notebook. In our next video, we will go to practical 1, that is the complement system. So before you perform it yourself, watch it before the lab session, and good luck. Remember, every great immunologist started exactly where you are now, in the lab. So learn to observe, to question, and discover. So see you in the lab next week. Thank you. Okay, and here are a few references for your knowledge. Bye.